Welcome to Wadham's webinar on COVID-19 trials and testing, principles in pharmacology for the clinical provider, presented by Shawn Smith. Thank you for taking the time today to join us. I'm Sarah Vega, the program assistant for Wadham. I will cover some logistics before we get started and introduce Shawn, and then help facilitate the question and answer portion near the end of this webinar. During the webinar, feel free to submit questions to the presenter via the question tab in your control panel, and we will share your questions during the Q&A session. You may also use the hand raise function on your GoToWebinar control panel to verbally ask your question once unmuted. In addition, we'll let you know when the recording and slide decks are available via email. Now I'd like to introduce Shawn. Shawn Smith is a US-based humanitarian flight nurse paramedic who has practiced and taught advanced critical care and emergency medicine from neonates to geriatrics for over 20 years. Shawn's career highlights include his service with the U.S. Navy, deployment on 50-plus medical missions, including the Hot Zone during the 2014 Ebola outbreak, and the PRN medical program support to the NASA Space Shuttle program. He is currently providing COVID-19 clinical guidance and training to numerous agencies, charities, and hospitals at home and abroad. Shawn's medical mission work has given him firsthand experience with many contagious or uncommon pathologies such as Ebola, diphtheria, cholera, rabies, leprosy, and more. He holds degrees in molecular biology, business administration, and nursing, and numerous postgraduate certifications in tropical medicine, disaster humanitarian response, and is currently completing a master's in forensic science. His education and experience have provided him with extensive familiarity with the molecular biology and rapid diagnostic testing strategies used in the current and associated projects management of SARS-CoV-2 associated pathogen. Oops. Thank you for presenting today, Shawn. I'll hand it off to you.
No worries, no worries. My goodness. Gosh, anyone that knows me knows I'm not into that. That's why we put it on the website, is so that you won't read it and embarrass me, but that's okay. That's okay. No worries, no worries. I always joke that, you know, my name tag just says Smith, and I tell people I'm the janitor. All right, but yes, I have done a few things related to this current outbreak here and there. Again, first off, I'd like to say thank you, of course, to Wadham for hosting the webinar. Thank you so much, Sarah, for that introduction. I'll have to spend the rest of my life living that down. And then, of course, going back over 20 years, thank you to all of my molecular biology and immunology professors, as well as my current forensic science professors. Go Gators. A small thank you, of course, to Abacus Pharma International. We're going to be doing a live rapid diagnostic test that they graciously provided for us today. And of course, thank you very much to all of you who are taking time out of your day and no doubt hectic work schedule to listen in. As Sarah said, all of this will be available afterwards. You'll get an email. If you registered formally, if you're not just looking over someone's shoulder, if you registered formally, you'll get an email with the slides and the recording. Okay? And it'll also be available on YouTube. And of course, as always, I'm available for specific questions, either within the context of the webinar, or you can reach me via email, Facebook, etc. Afterwards. So disclosures, in terms of financial disclosures, I don't have any to make, but with regards to the rapid diagnostic test, the RDT that we're demonstrating, again, Wadham's not specifically endorsing it. We're not in the business of doing that. And there's many, many tests out there. This particular test, I happen to be fortunate to know the CEO and the medical director. We've worked together off and on in Haiti for 10 years. Okay? Contact information and website for that is there on the slide, covid-rapid.com. And of course, we'll be using the API COVID Rapid test. My contact information up there on the right, my email, criticalcareprofessionals@gmail.com. You're also welcome to find me on any of the various social media platforms, and that is my profile picture that I commonly use. All right, so there is just an absolute avalanche of information out there, and Wadham in particular has actually been ramping up in terms of how we're filtering and curating that information for the disaster and humanitarian practitioner. So what we are going to talk about is we're going to do some basic molecular biology and biochemistry for different agents that are currently being used or trialed. Very briefly touch on some of the challenges of developing a vaccine. That's going to be super briefly because Wadham just did a spectacular webinar on that with Dr. Jerome Howard, and I'll show you how to get to that. Additionally, and one of the big things we'll focus on are the current tests. We'll go over how polymerase chain reaction works, as well as various immunoassays, and particularly the RDT. Okay? And talk about the science, the pros, the cons. What are your takeaways? I always say theory is utterly useless if it can't be translated to the bedside, right? So it doesn't do any good to talk about molecular biology if you can't supply the, so what? How does this impact my clinical practice? What does it mean to me? How can I use this information? Okay? What we're not going to talk about is I'm not going to go into any specific trial. I'm going to show you a slide in a moment and show you that there are there's over 2,000 clinical trials going on right now. And honestly, the amount of misinformation out there and things taken out of context it's just mind-boggling. I probably 90% of what I see, even in quote unquote science or popular science publications, etc., there's grotesque inaccuracies as far as how they're reporting on the actual science. And then many of the studies themselves have flaws, such as, for example, you saw recently that several studies were retracted, notably from like New England Journal of Medicine, with regards to hydroxychloroquine. And of course, the emergency use for that drug was just pulled day before yesterday. So we've been back and forth on things, right? Don't take your ACE-ARBs, don't take NSAIDs, this, that, the other. So the point is there's 1,000,001 things swirling around. So I'm not going to go study by study. We're doing broad brushstroke molecular biology to help you understand the why behind why certain things are being trialed or how certain tests work. Okay? Any one of these therapeutic or testing options, as I said, we could spend an entire day on the molecular biology of it. And honestly, it breaks my heart, guys. I mean, like we could just talk about IL-6 inhibitors all day long, right? And then downstream JAK inhibitors and, how interferon and tumor necrosis factor alpha play into that. We could easily spend an entire day just on that, or just on antigen antibody testing. I mean, gosh, I did 3 semesters of graduate school work on nothing but that. So this seminar, again, broad brushstroke molecular biology with respect to treatments and testing. It's to plant seeds of inquiry in the garden of your mind, point you in the right directions by curating resources. As time provides, we'll answer selected questions live. And then, as I stated, I'm very available to you offline. Because of the deluge of misinformation, I have deliberately limited the resources that I've made available to you. I'm curating, I'm giving you the best of the best in my humble opinion. As we just said, there's tons of stuff readily available on the internet. The problem with the information superhighway is you don't have to have a driver's license to get on it. I've given you screenshots in the body of the lecture. Several organizations that I recommend outside of those screenshots, and those screenshots mostly refer to NIH or CDC, FDA, things like that, which tie back to the World Health Organization, because this is an international lecture for an international audience. Professional associations I would recommend if you keep up with, Society for Critical Care Medicine, of course, Wadham, and AACC, formerly known as American Association for Clinical Chemistry. That last one is particularly good if you want to really dive deep on the molecular biology of testing. I do use selected infographics from JAMA, BMJ, New England Journal of Medicine. I think one other one that I cite in the body of the text. And then, because I know this is a broad audience, and I'm trying to make it broad brushstrokes, because as I said, it'd be very easy to get down in the weeds with one particular thing, like a JAK inhibitor or something. I've actually used a layman's site, but it's quite good. Okay? Compound interest. I see there I've spelled the daggum name wrong. It's compoundchem.com. So my apologies for the spelling error there. Compoundchem.com. Spell check just isn't going to catch that because it already sees it as spelled wrong. That'll be fixed in the final slides. Another thing that I like about compoundchem.com is not only does it translate the science into coherent language without dumbing it down, but it's also extremely multilingual. So again, as this is we have a global audience, it's nice to have a resource that's multilingual and reaches everyone from a layman to a molecular biologist. So these are going to be the primary things that we rely on. Again, a recent study showed that all recent studies are false, right? Garbage in, garbage out. We see tons of bad stuff with regards to COVID. And I'm sure if you, without even trying very hard, you can think of several things that have already been, that were the latest rage, and now they've already been discredited. And again, if you read, listen, watch with a skeptical eye, many of these things, people are not going to jump on the bandwagon. For example, in early March, I did a webinar for Wadham, and in early March, right, everybody was saying, oh no, don't use non-invasive positive pressure ventilation. And I spent, a good 10 minutes talking about why non-invasive positive pressure ventilation was actually something that should be very strongly considered in COVID. And now, of course, it's actually the official recommendation for precisely the reasons that we discussed. So again, try and be proactive and science-based versus reactive in reading other people's, pablum, their digested version of the literature. Get in the literature and read it for yourself, and read it with a very critical eye, because there are a lot of studies out there that they should raise questions about their validity. Okay? It's one of the bad things about the world of COVID is that we bypass the peer review process, which honestly, even in the normal world, isn't stringent enough in my book. Okay? Some of the things that I see get published, honestly, my old professors would have thrown me out of their office if I had dared show up with such garbage. So again, skeptical eye. All right, so rapid diagnostic test demonstration. And I'm going to go over that for you real quick. And the URL that you see here, that is actually my colleague, Dr. Vince Gennaro, demonstrating it. I'm going to demonstrate it for you live. And what I'm showing you here on the bottom, and we'll dive into this a little deeper later. This is just kind of the broad brushstroke interpretation, and you'll see there's a lot of ifs, ands, and buts. But what you're going to see when I do this, let me get my mouse over here, right? We're going to put a drop of blood here, and it's going to move, right? It's basically like chromatography, right? It's going to diffuse through the plate inside this plastic shield. And as it does that, right, it's going to bind that serum. If I have antibodies in my blood, it's going to bind antigen that's also bound to gold particles. Okay? If now the control line, it's going to migrate all the way through to the end. And that's if I have no antibodies at all, right? Then if I have IgG, oh, sorry, let's, not sure what happened there. Let's try that. If I have IgG, right, you'll see I get a line underneath that. And if I have IgM, I'll get a line underneath that. Ways that this can be interpreted. Obviously, There can be sampling error, or I cannot seroconvert, or referring to that diagram on the top, which I'll blow up for you later and show you in greater detail, is you produce, just like the viral genetic material, right, peaks and wanes, your antibody production peaks and wanes at different times. So if you happen to do your test and you're not at a peak period where the serum levels are within the threshold of detectability, you're just going to get a control line. Or that's if you had no antibodies at all. Or, for example, you could have IgM, but you could be early or late relative to the IgM peak, and it wouldn't show up. Same thing with IgG. Okay, and we'll go over that timeline later. But for now, I'm just going to show you very quickly how to do one. And it takes a few minutes to develop, so we'll do it and set it off to the side. If you'll excuse me for just a second, I'm going to escape out of the slides so that I can see myself in the video camera. Okay. All right, good deal. And yeah, let's turn that down a little bit for you so that you can see my hands. And let's see, so I've got my test here, all right? And as I showed you in the photo earlier, all right, boom, boom. All right, so this is a single test. Got a pipette, lancet, alcohol swab, and of course, the reagent. Now, another thing to keep in mind with regards to these tests is it's very important to have things at the proper temperature control. For example, when I was in Liberia, I worked and taught in the hot zone, and so all of the officers from the United States Public Health Service Corps had to go through my hot zone training. Well, they were using iStats. Wonderful, wonderful device. At one time, I personally owned like a dozen myself. They're great for that sort of work. Well, their iStats weren't working, and they were going nuts trying to figure out what it was. They had done all the troubleshooting, gotten on the phone with Abbott, etc., couldn't figure it out. Well, the problem wasn't with, in this case, it wasn't with the reagent, it was with the CPU of the machine. It'll only function within a certain temperature range. So if it's too cold or too hot, it's not going to work. And that's what was driving them crazy, because normally it's your reagents, right, or your test itself that's bad because of temperature extremes. In this case, their cold chain, everything was totally perfect, but they were leaving the CPU, the processing unit, sitting out on the counter, which of course in the African heat, it literally, was frying its brain. So it's like, oh yeah, just take that thing and throw it in the fridge. It'll probably be good as new. And they're like, Smith, are you freaking kidding us? Like, no, seriously, take it, throw it in the fridge. Do this all the time in Haiti. And sure enough, put it in the fridge. And 10 minutes later, they had a functioning iStat and were able to do tests on all their patients. Okay. So same thing with not just your processing unit. Now, this doesn't have an external processing unit. That's one of the advantages of these RDTs, right? Is they're not equipment or technology intensive. They're not even time intensive. You're going to have your result literally in a matter of minutes. Okay. So again, the reagent and the test itself have to be temperature controlled. Typically for stuff like this, because we do use it a lot in forward deployed medicine, room temperature. It doesn't necessarily have to be kept refrigerated. Often that's not a bad idea, but often you're in places where you don't have electricity and it's hard to maintain a cold chain. All right, so we've gone over some of that stuff. Let me open this thing up for you. Give you a look at that. Okay, so what's inside here? We've got the test, just like you see there. You see how that correlates to the slide, right? If I turn it like that, and let's see there. I'll put it down so the light's not right on it. So you can see the well, you can see the diffusion tray, the marks for the different types of gamma globulin, okay? And I should mention, right, of course, that IgM is generally, that's going to be your first one, first immunoglobulin that you form, and it's usually, you see that with an active or waning infection. And then, of course, your lasting immunity is the IgG, which you'll see later in the timeline, okay? So when we look at this, in theory, if it pops positive for IgM, that could mean that I have an active infection or a recent infection that I'm recovering from, okay? This is just desiccant, right? Heat and moisture, okay? So, boom, boom, cleaning off my finger, right? Got my land set here, right? All my diabetic friends know what's going down now, right? And then, of course, right, some area where you don't have a callus. So for me, that's usually like on the side of a pinky. And of course, I get a nice big drop of blood. I've got a clean pipette. I'm loading the well with the blood. I'll show it to you in just a moment. It's just a little difficult to try and do that up where you can see it. Nice big sample for you here. Hopefully not too much. Okay. So you can see there's blood in the well. I don't want to turn it too much. And then I'm going to take my reagent. Okay. First, I'm going to apply a little direct pressure. All right. Stop that. You can tell I've been eating a lot of green leafy vegetables. Okay, got my reagent. All right, and of course, this just opens up. All right, boom, boom. I'll put a couple of drops into the well. 2 drops of blood. All right. Some reagent. And I'll show it to you in just a moment. I'm giving it a chance to completely diffuse into the tray. Get this stuff out of the way. So, let's see if I can, there, if I tilt it, you can see it's moving up. You see the white at the top and the purple at the bottom. Okay, so I'm going to set that off to one side here and let that develop. Okay, and we'll talk. See, now you're seeing my private sanctuary, right? Nikola Tesla in the background, Tigger, all kinds of good stuff, right? Helmet bags, etc. Okay, let's go back here. Go back to presentation mode. All right. So we've got that test developing, and we've explained in general what the results could possibly mean. Okay, if you don't see anything but a control line, assuming that nothing's wrong with the test, that either means that I have never been exposed to coronavirus antibodies, or rather coronavirus antigens, so my body has not made IgM or IgG. If we just see, or that could also mean that I've been exposed, but it just happened. So my body hasn't had time to make the antibodies. Okay? If we just see IgM, that could mean I was recently infected. And of course, if we see IgM, or rather IgG, that can mean I'm out of the window of active infection, but that I had a past exposure. So it's one of those bad news, good news things, right? The bad news would be that I had coronavirus, but I have a high enough antibody titer of immunoglobulin G to gamma globulin to pop positive on a test. So, yay for me. I can have convalescent plasma, right? Or help develop tests and therapies for other COVID victims. Now, you can't see it, but right now it's only showing a control line. And having done a few hundred of these for various, more than a few hundred of these for various diseases, highly likely that's all this is going to pop positive for as I'm watching it develop. I'll show it to you in a few minutes. Okay. Boo-hoo, I wanted to be loaded with gamma globulin. All right, treatment. So I am showing you a central US reference, but global scope, okay? So if you go to cdc.gov, they have rearranged their website, okay? So now what you're gonna see is you're gonna get two hyperlinks. This page used to tell you what clinical investigations were going on. Now it simply links to the most current guidelines, that's the top link, and then most current clinical trials, that's the bottom link. Okay, here's the clinical trials, and I told you there was over 2,000, right? 2,172. So you can understand why we're not going to dissect these out in particular. I always say what you memorize, you can forget. What you understand, you know forever. So what I'm trying to do with this lecture is kind of give you the keys to the kingdom or the Rosetta Stone, even if you've never done molecular biology or immunology. Now, if you're a whiz at it, this will probably be a little boring for you, but hopefully I'll give you some tools that you can teach with. And if it's new to you or you're not a whiz at it, then hopefully this will get you up to speed and give you tools, right? Special forces, right? You always talk about a force multiplier. So it's always, you know, each one teach one. I teach you, you teach 10 people, they teach 10 more people, etc. Okay, so clinical trials, you can see over 2,100 studies. Rather than dissecting out a bunch of studies talking about strengths, weaknesses, pros, cons, we're going to talk about principles, those underlying principles, you know, that tie the movements of the symphony together, right? Things that'll hold true throughout. If you click on the NIH link, it takes you to this page. It's pretty easy to remember, COVID-19 treatment guidelines.nih.gov. Okay, and I just want to show you what the landing page looks like. And then, of course, when you get there, you click on View Guidelines, and this is what you'll see. And of course, this was just updated yesterday, right? Showing that we revoked the emergency use authorization for chloroquine and various derivatives. Now, that being said, if you're currently in a clinical trial, your doctor recommends it, etc., you can still get these drugs. Okay? It's just they're no longer covered under the emergency use authorization. Okay? And later I'll show you why people thought that these were a good idea, as well as briefly touch on you know, some of the problems with them, some of the confounding variables. Okay? So that's one thing to point out there. And then the other thing to point out is what you don't see. Like, for example, you may be familiar with the chloroquine derivatives and their current status because that's plastered all over the news. Well, the other thing that's been plastered all over the news in the last 48 hours, of course, is what? Steroids, right? Dexamethasone, right? Using a low-cost steroid, or if you've been really following closely, right, there's an antacid that we'll touch on briefly. So some of those things won't be reflected here, or if you dive deep and you click on these links off to the side, it'll explain that, hey, it's under investigation, okay? But this tells you what is currently approved for use, okay? And the level of evidence, and etc. Okay? So we'll touch on, as you saw, right, there's different antiviral, immune-based therapy, antithrombotic, concomitant medications. We'll touch on a few of those. Okay? Antiviral therapy, some immune modulators, do a little pro and con on, you know, empirically anticoagulating patients, because of course COVID just tears up the endothelium. And if you remember, darn it, I probably should have included a picture in here. But if you remember Virchow's triad or the clotting cascade, right? The classic clotting cascade. In reality, everything happens in once, but the way it was discovered and the way it's classically presented, right? We break it into an intrinsic and an extrinsic pathway. The intrinsic pathway, of course, is damage to the vascular endothelium. So that's heparin, right? The extrinsic pathway, think about that like Coumadin. That's when blood hits something just to make it simple, blood hits something that it doesn't recognize as vascular endothelium. So this is why, like, for example, if you have a mechanical heart valve, you're on Coumadin for the rest of your life, right? Well, I shouldn't say that. There's some other drugs you could be on too, but you get my point, okay? We'll talk about some of those direct-acting anticoagulants a little bit later. The point is that coronavirus, this novel coronavirus, tears up the vascular endothelium, so it triggers what we classically think of as the intrinsic clotting cascade. So one whole area of research in clinical practice is exploring, DVT prophylaxis and dealing with this microclotting phenomena, etc. And then, of course, some other drugs. Now, I really shouldn't have put HCQ there, hydroxychloroquine, because technically it's kind of antiviral, but I put it in the catch-all because it actually has multiple proposed effects. Steroids would be another example of a concomitant reaction per concomitant prescription rather by NIH. Okay. Testing, again, US-based references, but global scope. Here I'm just showing you a screenshot of the FDA's page showing you what tests are approved in the US, what aren't, yada, yada, yada. How do you get that information? And again, we're very thankful to API for loaning us these tests so that we can demonstrate it for you. But, in terms of full disclosure and, conflict of interest and all of that, I want to give you this information as well so that you have access to all the various types of tests as a clinician, as well as a layperson, and as someone who's educating both healthcare providers and lay people. Okay. So this is the blow up. All right, this is the blow up of that little tiny slide there, a little tiny picture that was at the top of the slides. So you can see here, right, before symptom onset, right, you're unlikely to detect antibodies or viral genetic material. And you've got some different lines here. I'm going to show you a different graph as well that's a little less busy and perhaps a little easier to follow. But you notice PCR likely positive and then antibody detection. So you see you're going to pop positive depending on the threshold of the PCR test you're using, right? You'll probably pop positive for that before you pop positive for IgM. Remember, you're going to make IgM first. It's going to peak first. And then you'll pop positive for IgG, okay? PCR, polymerase chain reaction. So if you think back to your basic biology, right, when we talk about DNA or RNA, talking about a double helix, single helix, right, and we transcribe, right, we go through transcription and translation. So PCR is basically doing that in the lab, but doing it at an incredibly accelerated pace, where we basically take genetic material, break it into fragments, and then use other bits to make copies of it and amplify the production. So polymerase chain reaction. It's very gratifying to see what they're doing with this now, because I did it back in the day. I mean, hell, I still remember when the first paper came out, okay? And we had to do all of this stuff by hand, okay? And it was sensitive and finicky like you wouldn't believe, okay? But anyway, polymerase chain reaction. So spelling that out, and again, re-emphasizing IgM before IgG, okay? Here's another that I feel is perhaps a little less busy, okay? So asymptomatic stage, you see days on the bottom, right? And then, of course, your y-axis isn't labeled, but it's basically referring to levels of IgM, IgG, and, of course, viral genetic material. So we've only got 3 lines on this one. It's a little bit easier to follow, okay? So it's showing you there, right? And it's labeled which one's which. So red, green, blue. Shows you when one becomes detectable, when one disappears. So you can see the sweet spot's roughly around 14, 15 days where you might pop positive for all three, right? Your PCR might pop positive, your IgM, your IgG might all pop positive. And then of course, you can see that your IgM will be gone basically by the third week. Your PCR, assuming that you go through a normal therapeutic course, your PCR is going to be completely gone by the fourth week. Okay? And if you did get lasting immunity, lasting is open to interpretation. You know, everything's an unknown and a dynamic process with COVID. But what we're looking at is approximately 1 year. That's our best guesstimate for how long the IgG will hang out. Okay. All right. So things that can affect these tests: timing. We just spent a lot of time talking about the timing. Contamination, sensitivity, specificity, seroconversion, persistence. How long does it hang out? So timing and persistence. Cross reaction. Some of these antigens will cross react, and you've got two antibodies in play, so there's double the fun, double the chance of something to go wrong. What do I mean by all of these? Again, timing, we just went over that chart. Contamination, right? Could I get samples mixed up or use a dirty pipette between different samples, this, that, the other? It really depends on the collection methodology. How we do it in healthcare is a lot different from how we do it, for example, on a bench in forensic science. And both of them have similar but different quality assurance, quality control processes. In forensics, you know, someone's innocence or life literally hangs on the results of those genetic material tests. So we're, you know, super scrupulous about the whole chain of evidence, quality control, and quality assurance during the lab. You know, the personnel's certifications are constantly verified, the reagents, you know, periodically checked, etc. Healthcare, we have some of that in healthcare, but of course, in the time of the pandemic, everything's being slammed and rushed. Okay, that's exactly how we get some of these garbage papers. Seroconversion. There's always the possibility you can be exposed to the virus and not have a strong enough immune response to produce the antibodies at a level that'll pop positive on the test. I sometimes have a problem with this in hepatitis B, and it's really interesting because, I mean, I've been, as Sarah told you, I mean, I've been exposed to damn near everything and had, you know, I mean, gosh, just back in March. Well, anyway, point is I've been exposed to an awful lot of stuff, some of which is incredibly contagious. I've received vaccines against everything under the sun, etc. So it's always kind of interesting that I don't seroconvert so much for Hep B. And I'm looking at my test right now. You can't see it, but as I predicted, only the control line is showing. So one of the possibilities there is what? That I haven't seroconverted. I could have an active COVID infection. I could have a past SARS-CoV-2. I'm just saying COVID for easy math. Please excuse my sloppiness. I hope everyone understands the difference between the disease and the virus. So I should be more precise in my speech. So I'm just popping positive for the control line here. So I could have an active infection, I could have a past infection if I didn't seroconvert. That's one possibility, okay? Persistence, again, how long does it hang out? How long does the immunity hang out? Cross reaction, for example, previous exposure to a different SARS coronavirus, okay? And that's just a hypothetical example. Again, two antibodies, so depending on the immunoassay, there's possibilities for error there. Generally speaking, IgM has been shown to have a lower sensitivity and specificity. Okay? What do I mean by sensitivity and specificity? Sensitivity, of course, greater true positive, less false negative in terms of results. specificity, greater true negative, less false positive. Clinical take home, something that drives me nuts in the news, reinfection. Oh, I got COVID twice. No, it's highly unlikely, right? Very, very unlikely. You may have heard, I say Occam's razor because, again, I took 5 years of Latin, etc. Was trained in classical logic and rhetoric. Occam's razor, the simplest hypothesis, or as we often say in medicine, when you hear hoofbeats, look for horses, not zebras. It's highly unlikely that someone's gotten reinfected. What's more likely is that there's errors in their testing procedures. They didn't get the swab back far enough, so they didn't get enough sample to pop positive on a test, for PCR, etc. They had washout or tailing on their antibody-based test. So they popped negative once and positive again, so again, reinfection unlikely. Okay, hit me up offline if I didn't make it clear why. Here's the time for me to share the result with you. Again, I'm just going to escape out of here for a moment. You guys can actually see both. You can see the slides and the video screen. I can only see the slides. So I'm literally flying blind here, but no one's raising their hand or telling me I'm making an ass out of myself, so I must be doing okay. All right, let me escape out of here, go back over here, and I just want to hold this up for you. And again, I'll have to tilt it. I'm trying to find the light. There. Okay, so you see the M on the bottom, going from the bottom to the top. You see the M, then the G, and there's no lines next to those. So where do you see the line? You see the line up here at the top in the control. Okay, so basically that means that my blood and the reagent migrated all the way through this without binding to any antigen that was attached to gold particles. Okay? So it went all the way through to the control strip. If I had had antibodies, depending on the antibodies, I would have multiple lines. If I had a current infection, I might have a line at the M line, IgM. If I had an old infection, but I had seroconverted and achieved immunity, I might have a line at the G line, okay, for gamma globule. All right, so just showing you real quick how one of those works. If you've never seen one before, hopefully that was instructional for you. All right, let's go back to, now return you to your regularly sized slides. Well, so how do these tests work? We're going to blast through some stuff here in about 10-15 minutes and go through different testing. Again, all of these graphics, British Medical Journal, New England Journal of Medicine, or JAMA, unless I tell you otherwise. Okay? And if I tell you otherwise, it's generally cited in the slides. So how does PCR testing work? So you see that swab, and again, going all the way back to the nasal turbinates, right? So you can see actually, just whoa, that's really deep, right? So you can see right away where there would be a potential for sampling error, okay? Then we take these primers, right? Like you may hear someone use the term Taq polymerase, okay? So we've got primers and then polymerases, right? Polymerase chain reaction. So if you ever hear someone say Taq, they're talking about a specific polymerase. See me offline if you want to get freaky geeky with it. So I'm duplicating it and then I duplicate those, right? So it's logarithmic. It's kind of like bacterial growth, right? It's exponential. First I have 2, then I have 4, then I have 16. So polymerase chain reaction. So amplification creates millions of copies. And what we do here is that genetic material, we incorporate a fluorescent marker. And there's all kinds of ways you can do this. Shame on me, I really should have patented one I came up with almost, 30 years ago. You guys have all heard of bioluminescence, like, in the ocean, etc. Organisms can be bioluminescent, etc. Well, there's a bacteria. Now, Sarah said at the beginning of the lecture, she mentioned that I had worked with cholera. Well, that's from Vibrio cholerae. Well, Vibrio, there's another Vibrio species that is bioluminescent. So what you do is you take something like that and you clone that gene in. And so as you amplify it, of course, the more, that's what I did all those years ago. That was the particular source that I used, and was working with Haemophilus influenza. So basically, when you look at the fluorescence, or whatever tag you're using, in this case, bioluminescence, the brighter it is, right, the higher the concentration. And you can literally create a concentration curve where I get this much luminescence, it equals this much load, okay? So positive result, if there's enough genetic material in the sample, application, and the fluorescent markers are high enough to be detected. If there's not enough, too low to be detected. Now, there's a million and one things that can go wrong with this. Again, we could spend an entire semester doing the ifs, ands, or buts, but this is the basics. Okay. Okay. Here, this is from the one I showed you before, compoundchem.com. Compoundchem.com. Okay. And so here, showing you how a PCR works. This is the exact same test, just a different representation of it. And what I'm going to do is take the important parts of this and blow it up on individual slides for you, so you can see it a little more closely than maybe you could have on the last slide, or even this one. So here you see I take a swab inside of the nose, back of the throat. Okay, I've got viral RNA. Okay, we extract the RNA out of the virus. You see the intact virus there, right? We extract that, we purify it, we get all the garbage out, because we don't want our primer, right, our primer and our polymerase to start binding and amplifying garbage. So we purify it. We use a reverse transcriptase. Remember, I used the terms transcription and translation, right? So normally, like with DNA, what do? We transcribe DNA to RNA and then translate RNA to protein. Well, here, because this is an RNA virus, we have to convert the RNA to DNA. So instead of using a transcriptase, we use a reverse transcriptase, because we think of, right, our normal order as being DNA to RNA. That's how it works in us. But in this particular virus, it's an RNA virus. So we have to use a reverse transcriptase to convert RNA to DNA. Okay? DNA is mixed with primers, right? And heat and cool, right? And this was the really difficult part when we were doing this from the very beginning back in the day, right? This is the crucial part, is it has to be kept within a certain temperature range. So we heat and cool, DNA building enzyme, polymerase. So polymerase chain reaction, the P, that's that DNA building enzyme, right? Millions of copies of the viral genetic material, okay? And then that is bound to these fluorescent dyes. So the more copies you have, the brighter it is, right, when you're assessing it, okay? Boom. And again, you do a whole calibration curve for this. That in and of itself, takes, a good week to go through how to explain that. But once you know how to do it, it's super easy. But the theory, I just gave you the theory. Fluorescence increases as more copies of the virus are produced. You cross a certain threshold, positive. Doesn't cross the threshold, negative. Okay, and you see there fluorescence versus copying cycles and the threshold line. Okay, and again, this is compoundchem.com. Okay, some possible issues here: supply and demand on the reagents. And this also takes much longer, right? You saw the one that I just did. We did it in a matter of minutes, okay? False positives and negatives, right? If the sample degrades or gets contaminated, we're going to have issues. And I explained how that comes into play with forensic science in the courtroom, okay? As we make them prove, right? Or in my case, we have to prove, okay? So antibody test, that was a genetic material test, your polymerase chain reaction. Let's look at some antibody testing. So here's part one. I didn't blow anything up on that. That's the theory that I already went over verbally with you. Okay. Antibody test part 2. So we're going to look at a few of these very quickly. They essentially all work the same way. It's an antigen-antibody interaction. And if you look down at the bottom there, you'll see a bunch of things that look like Y's. Those are your immunoglobulins. Those are your antibodies. So Ig, immunoglobulin, so like IgG, immunoglobulin G, IgM, immunoglobulin M. So I got my little Y's down there, and those bind, right? Those bind antigens, okay? So all these tests basically work on the same principle of an antigen-antibody interaction, okay? So with an RDT, that's what I just showed you, and you can see how this directly correlates to what you saw, right? You see the sample going in on the left, and what's in that sample? It's a drop, and they're showing you some antibodies, okay? That's assuming that there's some there. My test came out negative, so I didn't have enough antibodies. There's antigen bound to gold nanoparticles. So if I have antibodies, they'll bind to that antigen as they wick. It's just like water moving through a paper towel, right? Or if you do trauma, you're familiar with the halo test, right? If I have otorrhea, right, CSF, and I touch some gauze to it, I'll wind up with a red dot in the center and a yellow, red for blood, and then yellow around that for CSF, because the different fluids have different molecular weights, so they move at different speeds, okay? So here you see it migrating up. This particular one, if it correlated directly to the one that I showed you, this guy would pop positive for IgM and IgG. You see the 3 lines there. So that means he's at the tail end of an active infection, right? Whereas IgM is starting to fall, but his IgG is starting to come up. Okay. Again, one of the big advantages about this, very quick, very cheap, very low tech. Anyone can do this, right? I can train someone very quickly on how to do this. So a community healthcare worker can do this. Okay. Again, it's just going to say yay or nay. It's not going to tell you how much antibodies you have or how effective your response is. Eliza, very similar. In this case, Eliza, RDT stands for Rapid Diagnostic Test. Eliza stands for Enzyme-Linked Immunosorbent Assay. Enzyme-Linked Immunosorbent Assay works the same way. We're just using enzymes to cause a colorimetric reaction. So instead of using the gold particles to form that line where it kind of almost precipitates out, right, you're doing a colorimetric reaction. So the PCR was a fluorescence type reaction or a luminescence reaction. This is colorimetric. So the more intense the color, the greater the concentration. So you see when you look what this test can tell you, it's because it's a colorimetric assay, right? It'll tell you, do you have antibodies, yes or no, and what is the level of the antibodies? But of course, it's not going to tell you how well you're fighting off the infection. These others, I'm not even going to dwell on them, just variations on the same theme, okay, where the antigen-antibody complex is bound to something else. And then you use that something else to quantify how much antibody is in a given volume of serum. Okay? Boom. Big things to take away with rapid diagnostic tests, right? Why are rapid diagnostic tests so important, especially in something like COVID or even Ebola, right? Malaria, things like this. Well, with COVID, the big deal, and again, forgive my slang, but it's just easier to say COVID, right? We have patients who are asymptomatic, so and they're transmitting, right? We're worried about asymptomatic transmission. So RDTs can help identify positive individuals who are asymptomatic, assuming they're in the window and they seroconvert. It's not resource, time, or technology intense. And of course, it also identifies people that can be useful for treatment, right? If you pop positive for IgG, you have immunity. So now you can take care of COVID positive patients with minimal risk to yourself. And the only thing you have to worry about, of course, is deconning when you leave so that you don't carry it to the outside world. And of course, you could also be a candidate for convalescent plasma. Okay. Something, just an aside I threw in here, this just popped the other day. Again, it's a minor, it's an observational, anecdotal case study thing, but it was kind of worth writing up. So I put the write up here. Basically, they thought that these patients had COVID encephalitis, so they checked their CSF. Okay, and remember, IgM doesn't cross the blood-brain barrier. So if they pop positive for IgM, that means that the virus had to be on the bad side of the blood-brain barrier. Okay? So they found that these patients whose CSF results were otherwise unremarkable, like often with viral, you may see elevated protein or elevated lymphocytes if you have viral meningitis or encephalitis. These patients were negative for that, but they popped positive for the antibody. So something to think about with suspected COVID encephalitis, right? Little clinical pearl for you there. Other things to think about when you're looking at drugs and do they work, do they not work? Did this patient die of COVID? Did they not die of COVID? Remember that a lot of these drugs, like the chloroquinines, for example, they block potassium channels. So when you see the cardiac issues, the question is what's the patient's potassium level? And of course, then that ties into things like what's the patient's pH, what's their mag, what's their phos, the magnesium and phosphorus, sorry. All of those things are interdependent. And then, of course, we have to look at PD and PK, right? Pharmacokinetics, pharmacodynamics. How are the drugs metabolized? How are they eliminated from the body? Okay? So important things to look at there. Whenever you're looking at these results that say a drug works or it doesn't work, you need to look for confounding variables, right? Or it says, oh, hey, this drug causes arrhythmias. Well, did the drug cause the dysrhythmia, or was the patient, they had diarrhea and vomiting and poor PO intake and their potassium was in the toilet, and the drug wound up being to the back of their head because their potassium was already literally in the toilet. Okay, so things to think about when you're looking at some of these studies, okay, is ask yourself what else is going on with the patient and how well they standardize the patient population. All right, very quickly, I know we're over time. See, Sarah, that'll teach you to give me a big, long introduction. Next time, just say I'm the janitor, right? That would have saved us 5 minutes, bless your heart. All right. So we'll very quickly run through some drugs. Thank you so much, guys, for hanging out. I apologize for stealing a few extra moments of your life. We'll just touch on some of these things very quickly. And again, I'll be very available to you afterwards. Why did we use chloroquinine and its various permutations? Some of the proposed mechanisms of action the hypothesis, of course, is that coronavirus gets in through angiotensin converting enzyme 2 receptors. Chloroquinine may block that. Chloroquinine alters cellular pH, which then has downstream effects. Could also interfere with viral assembly, and it downregulates tumor necrosis factor alpha. You guys, and there I wrote a cytokine. Why did I write a cytokine? Because you probably all heard of the cytokine storm, right, in regards to COVID, right, that hyperimmune response, hyperinflammatory response. Antiviral example under current investigation, right, remdesivir. And this is really cool. I love adenosine analogs. In fact, I know I've got someone listening in the audience right now that owes me a homework assignment on an adenosine analog. And remdesivir, of course, adenosine analog, interferes with RNA polymerase. Think about DNA, right? Think about your purine, right? Gattaca, right? Adenosine, right? Think about your base pairs, right? So remdesivir, right, mimics the adenosine nucleoside and interferes with RNA polymerase, so it terminates the chain, right? So you can't have that transcription, it stops synthesis and then decreases the RNA production. This was another cool one that just came out earlier this week. Famotidine, you may know it as Pepsid-AC, serum protease. So this interferes with the viral replication. Low cost, PO, can give outpatient. Small observational study, but potentially exciting. Again, notice the golden themes. What are we doing here? We're either stopping the virus from getting in the cell, or we're interfering with the replication and assembly of the virus. If you think about HIV and think about how the therapies for HIV evolved, you'll realize these approaches are classic. Okay. Other antivirals, again, I'm just refer you to the NIH website. Vaccine, active immunity, right? I give the vaccine, it stimulates the body to make immunoglobulin. Issues that we have with vaccines, right? Getting it specific enough, the virus mutating, making sure that we ensure equitable access to all. So like a drug like Pepcid, that's low cost, right? Dex, a steroid, low cost, we can make sure everyone gets that. Vaccine, how do we make sure everyone gets it, okay? So again, Wadham has an excellent podcast on this, Professor Jerome Hauer, excellent, shameless plug for Wadham, go watch our podcast. Convalescent plasma, this is passive immunity. You're literally transfusing the patient with gamma globulin, okay? In addition to transfusing the patient with gamma globulin, other plasma components that may have anti-inflammatory, anticoagulation benefits. If you're taking notes, do jot this down, because shame on me, I didn't jot it down. Anytime you're doing something like this, of course, you run the risk of immunomodulated negative reactions like taco and trolley, right? Transfusion-associated circulatory overload or transfusion-related acute lung injury. Okay, so you know, it's not well characterized, but just be aware that's potential hypothesized complications of convalescent. They're just giving you a graphic showing you what I said verbally. Immune modulators. That's just the quote for this, the citation for this picture. Why am I going over this? Because steroids and IL-6, IL-1, IL-6, JAK inhibitors, this is a hot area of research, okay? So I'll let you look at this on your own in the interest of time, because we're already over time, but you can see the viral response phase, host inflammatory, and how they evolve over time. So what you want to do, or like down here at the bottom, you see, when do you give what, right? Here's my remdesivir, chloroquine, right, convalescent plasma, and it's showing you when you do what. Like, for example, you don't want to give steroids early on. You want to give them later, right? Or you don't want to inhibit the interleukins early on. You want to do that later. So this shows you, and so you've got the symptoms, how the symptoms evolve over time, how the clinical signs evolve over time, your lab values, and then what therapies. The problem that you see a lot with COVID is people want to do this fire and forget silver bullet stuff, and the natural history of the disease state evolves. So different therapies are appropriate at different points in the disease state progression. And that's why it's so important to understand the clinical progression and how the lab values change. Because if you give the wrong therapy, right therapy at the wrong time, you're going to violate the first rule of medicine, of course, first do no harm. So this, when you get access to these slides, or if you email them to me, print this one out, put a gold star next to it. Very useful for helping you understand why we give, what we give, when we give it. Okay? Here I give you a whole bunch of examples of immune modulators, right? So yeah, boom, 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 tons of them. I'll let you read the slide yourself. Another thing we can do with those circulating immune modulators is literally filter them out of the blood. Okay? Cytokines being a big target. Okay? Steroids, remember that your NIH guidelines, it's a reactive process. So when you look at the NIH guidelines, you're not going to see anything about steroids. But if you look in your inbox or look on the information superhighway, of course, everybody's saying, oh my God, the last 48 hours, steroids have become a miracle drug. Guys, there's problems with the study, this, that, the other. Okay, I'm just going to quickly give you the takeaways. Seriously ill COVID patients, 20 to 30% decrease in mortality. Generally, our vent-dependent patients see better outcomes from taking the steroids. Okay, and again, the problem here is that there's other studies showing varying efficacy with different doses and timing. Okay, so the jury's still out, literally. And this paper and this study, remember, in the physiology world or experimental world, physics, physiology, right, it has to be reproducible. So first we say, hey, is it real? Did you control for all the variables? And then is it reproducible? If so, this would be great because, again, it's a low-cost, readily accessible prescription and treatment, right? And that's what we're looking for here is lots of bang for buck, equal access for all, okay? Antithrombotics, I already went over the coagulation cascade a little bit with you. So questions we get, should we do increased VTE prophylaxis, venous thromboembolism prophylaxis on an inpatient basis? Because remember, you're working against, right, Virchow's triad, venous stasis and insult to the vascular endothelium, right? Plus you're hypercoagulable because of all those immunomodulatory effects. So should we up our VTE prophylaxis? Should we just empirically anticoagulate people? Those are some of the questions. Smith's law, you know, if you guys have ever been to any of my lectures, you've heard multiple Smith's laws, right? I'm like Murphy, I've got a million laws. This is the yo mama principle, right? If it was yo mama, would you want someone doing cookie cutter medicine, one size fits all? We're going to treat every patient exactly the same, no matter what their unique signs and symptoms and disease progression is? Or would you want yo mama to get individual treatment for her individual disease process? Okay, so right treatment, right patient. Again, I give you a link to an SCCM lecture on both immune modulators as well as antithrombotics. And spoiler alert, the conclusion they come to is yo mama, individual medicine for individual patients. Here it is, right here, COVID-19 pharmacology debates. It's already happened, but it's been posted, it's available. So you just Google that, Society for Critical Care Medicine, and it'll pop right up, okay? There's the panel for the immune modulators, panel for the anticoagulation. I just put those in there to show you the caliber of the people that they had discussed this. Okay, so this isn't some Mickey Mouse thing. You know, these are literally internationally known heavy hitters, not just in pharmacy, but specifically in anticoagulation, specifically in immunomodulation. So excellent. If you want to get down in the weeds on some of the current papers and studies, that's a great way to do it with respect to those two things. Sliding into home plate here, multi-system inflammatory syndrome in children. Again, no silver bullets, multifaceted, proactive, supportive care. Okay. In closing, again, thank you to Wadham, thank you to Sarah, thank you to Andrew, our support staff behind the scenes. Thank you to everyone that's, you know, tried so hard to pound knowledge into my thick skull and get past my traumatic brain injuries and all of that. And thank you one more time to Abacus Pharma International for providing us a test to demonstrate live. And thank you so much, guys, for not only taking time out of your day, but I can see the number of attendees and we haven't lost anyone. That's like the biggest compliment ever. I mean, I'm not getting paid for this lecture, but knowing that you guys are hanging out and you're getting something out of it when we should have been done quite a few minutes ago, that means a lot to me. So thank you so much. Again, look at things with a skeptical eye. Closing resources for you. Of course, if you do have any questions, comments, concerns, you hung out this long, I'll hang out a few more minutes. But if you have to go, you're more than welcome to hit me up on LinkedIn, Facebook, email, Twitter, Instagram. Quite honestly, email is your best bet. Okay. So again, I'll hang out for live questions. It won't hurt my feelings if I don't get any because you guys already made my day by showing up and staying late. If you do have questions and you got to run, hit me up offline. All right. Thank you so much, guys. I really appreciate it. I hope you got as much out of listening to me as I did out of you guys being here today.
Great.
Standing by.
Shawn, for that very comprehensive lecture. We do have some questions coming in. I have one participant with the hand raised. So in fact, she and I had been exchanging messages during the webinar, so I've encouraged her to ask you a question directly. So I'm going to go ahead and unmute Bonnie's microphone. Are you there, Bonnie?
Okay. Yes, I hear you. Thank you. Thanks for taking my question. My question is, if somebody had an active virus, like they had herpes or something, and came to have the COVID test, would any active virus cause, this test to be off?
Not the tests that I have shown. Okay. Neither of those. Sorry, I'm not sure why we're getting the feedback there. So I hope you can hear me okay, Miss Bonnie. And thank you so much. That's a great question, because I did mention cross-reactivity. But again, it's cross-reactivity is generally between the same type of virus, not always, but generally. So no, you're going to see very little cross-reactivity between herpes and COVID, okay? Or SARS-CoV-1, SARS-CoV-2. Okay, so did that answer your question, Ms. Bonnie?
Thank you. Yes, it did.
Okay, super. And again, if you think of something, we always think of questions after the fact. So if you think of something else, hit me up offline. But the short answer to your question is no. Next.
Thank you, Shawn. So we have a couple questions that have come in through the chat feature, and so I'll go ahead and read a few of those, but I will encourage everyone in the audience, if you'd like to ask Shawn a question directly, we can unmute your microphone and give you the floor for a question. So I'll go ahead and read a question I received here from Karen. I also tried to encourage her to ask the question directly, but she was content with me reading it out loud, so I'll go ahead and do that. Karen says, I'm seeing lots of clinicians do PCR sampling just from the nose, not far back, claiming that mucus is mucus, the virus will be there. Is that prompting more false negative results?
Potentially, yes. Absolutely. That's one of the biggest problems. Oh, and that actually brings up a good point. I'm glad you asked that question, because I always try and explain why. And you may have, one of the things you may have heard, is people having altered sense of smell, right, or losing their sense of smell. And this ties directly to your question. This is a case of form following function, right? Because there's a hypothesis when we talk about like COVID encephalitis, like I mentioned, testing the CSF for antibodies, that the virus is actually getting into that compartment through the cribriform plate, right, and up the olfactory nerves. So yeah, no, it's not. Snot is not snot, just to be blunt. Okay, you have to get further back, right? And that's one of the things that causes a lot of potential issues with this is sampling error when we talk about PCR. There's limitations to the rapid diagnostic test that I showed you, but it is a lot harder to get sampling error with one of these than it is with the PCR test for COVID. Other PCRs, we can sample different ways, but the one that we use for COVID, no, you got to get on back there, as we'd say in the South. Did that? And Karen, if that didn't answer your question and you're still too shy to speak up, just shoot Andrew another line. I'll be happy to take another swing at it. Thank you so much for that excellent question. It's a super relevant and important question. Thank you very much. Who's next, Andrew?
So we have a question from Stephen from Ghana, and he has two questions, so I'll go with the first one here. Stephen asks, what is the sensitivity and specificity of the RDT?
It depends on the RDT. I hate to give you a slippery answer, my brother, but it depends on the RDT, and honestly, it depends on how you define sensitivity and specificity. The one that I was using is advertised at, I believe, over 95% for both IgM and IgG. I would have to, you've got to get off into the weeds and dive deep if you want to validate that. But again, I've worked very closely with this medical director, and I know he does his homework. He's probably going to get on to me and say it's actually higher than 95, actually. Yeah, and that's another reason why it's important to look at the FDA website, Stephen, is because some of the RDTs that have been disqualified, it's because they didn't meet those thresholds. Right? So that's one way. You can't just say what is the sensitivity and specificity for the RDT, because it varies by RDT. But if you go to the FDA website, that'll give you more information, as well as the cutoffs. And if it's not consistently clinically accurate, it's not approved for use. Okay? And even if you're in Ghana, remember that this is a global problem. So we've probably tested the kits that you may be using. If you have trouble getting the information you need for your specific test, feel free to email me and I'll help you look. Okay? Absolutely. I don't know how the government in Ghana verifies, right, how they do their quality assurance, quality control. So my best hope is that the FDA, our Food and Drug Administration, has looked at the tests that you're referring to, and you can find the answer on their website. Okay? And if you see that it's not approved for use, that answers your question. All right, super. Thanks for that awesome question, Stephen. Andrew mentioned you had another one.
Yeah, Karen quickly sent a follow-up. She just indicated, thank you, helpful answer. So that was the question prior to Stephen's.
Okay.
Stephen also, and I think you may have covered this to some extent in the presentation. This came in a bit earlier, but he asked, are you not using HCQ in managing COVID-19? I believe that's hydroxychloroquine.
Yes, yeah, absolutely. And that's a totally accepted abbreviation. So I don't know if I mean, but yeah, HCQ. And I did touch on it very briefly. So it's in a state of flux right now. And that's what I don't want people to misunderstand, is the emergency use authorization has been pulled. But if you look at it carefully, that's all that's happened. In other words, if you're currently taking it, your doctor recommends that you should take it, you're part of a clinical trial, it's still going to be used. So the short answer to your question is absolutely yes, it's still being used. It's just that the emergency use authorization was pulled. Okay? I don't know how to make it any more plain. And there's differenements of opinion, all over the world. So there's still clinical trials ongoing with this. Absolutely. So great question. Thank you for re-emphasizing that key point, and also re-emphasizing how careful you have to parse out what you see in the media. Okay. All right. Hopefully that answered your question. If not, just keep asking. Whether you do it through Andrew or you email me or Facebook me after the show. What else have we got, Andrew?
We have just looks like one more question. We have several thank yous from the audience. I won't go through all those right now, but
yeah. No. I really appreciate it. I don't know if you noticed or not, but almost everyone has stayed. And I mean that's just phenomenal. I mean, I am very strict and very regimented with time, and if someone had kept me this late after a meeting, I wouldn't be happy unless I was very happy with the speaker. So again, you guys have my profound thanks and gratitude for hanging out. It's the highest compliment you could give me. Thank you so much. What was that last question, Andrew?
Well, suffice to say, a lot of positive comments, so that's great. So this is from Michael Pendon, and I'm probably going to mispronounce a few of these words here, so I'll just preface that. And Michael has two questions. First, he says, I've encountered some cases where patients had throat swab PCR negative while endotracheal aspirate sputum came in positive.
Sputum.
Okay. You can see I'm not terribly familiar with the medical terminology.
Basically, the patient was intubated and they suctioned out the endotracheal tube.
Got it.
And that sample then popped positive. Carry on. It sounded like he
goes on to say both tests were done on the same day. What could possibly cause this? Noted your lecture on factors that could cause false results.
Sure, absolutely. So any step of the way, there could have been a higher viral concentration, right, in the sputum, right, because everything's draining back there, so it's pooling. That's probably the most likely reason, assuming that both tests actually sampled accurately. That brings us to another potential error in that the first test could have not been done accurately, because you've got to break it down, right? I do a whole week-long series of seminars on lab values. I've edited a lab values textbook, and any time a system interacts with your patient, it becomes part of your differential diagnosis. For example, if a ventilator alarm is going off, your first question is it the meat or is it the machine? Is my patient broke or is my vent broke? Well, it's the same thing with lab values, right? If I get a freaky deaky lab value, the question is the patient broke or is the lab broke? And then when we look, is the lab broke? Every step in the process becomes suspect. So it could be a problem with how I obtained the sample. It could be a problem with how I stored the sample. It could be a problem with how I processed the sample. For example, they may have used too much diluent. Assuming that they got the first sample correctly, they may have diluted it out to the point that it wouldn't work, right? So there's a whole myriad of possibilities, and a lot of those possibilities are even unique to the equipment that they use, right? And that's what I meant earlier when I was saying, hey, you know, I'm not trying to be pedantic, but I literally just gave you like an entire semester's worth of material. I mean, more than a semester. I mean, I spent years learning a lot of these testing methodologies. So chances are that if I was a betting man, I would bet that when they obtained the first sample, they just didn't do it correctly. Whereas the second sample, because everything's pooling and concentrating, the second sample, it would have been almost impossible to do incorrectly. They were going to get a good sample. So I hope that answered your question. It was a great question because, again, sample error is a very important concept. If it didn't answer your question, just ask Andrew again or hit me up offline if you need to go. But thanks again for such an awesome question. Was that truly our last question, Andrew?
There was a follow-up, so it was a two-part question, also from Michael.
Okay, and I'm not in a hurry for me. I'm in a hurry for the people in the audience. So I'm happy to take the second part.
Sure.
Go ahead.
It's the last question.
Okay.
So I don't see more in the queue. Michael asked, is a throat swab enough, or does it need nasopharyngeal as well? We only do throat swab for PCR.
Again, assuming that you execute it appropriately and the viral load's high enough, a throat swab alone should be sufficient. And especially depending on how the patient, you know, there's things that can make it easier to get a high quality sample from the back of the throat versus the back of the nose. Okay? So yeah, throat swab's totally sufficient in and of itself. Another great question.
Well, that does indeed to be the last question for the session. So, and we just are right at the top of the hour here. So seems like a good closing point in the presentation today. As mentioned by Sarah at the beginning, this is being recorded, and we will upload the recording later this afternoon and share a link to that, as well as a link to Shawn's slides and an email address if you want to have a follow-up conversation or have any further questions. You can address those directly to Shawn by email. Did you have any parting words, Shawn, before we sign off here?
Nope. Thank you so much. Just stay humble and stay hungry, guys. I appreciate you all. Be safe out there. Stay safe, stay sane. Don't neglect your mental and physical health. Remember, you can't take care of the patients if you don't take care of yourself. Okay? Thank you so much. Really appreciate you all.
Thank you, Shawn. Appreciate it again for your time and sharing your expertise with us. And thank you to everyone in the remote audience. I know depending where you are, you could be early morning, late afternoon, evening. So we really appreciate you participating in the live webinars. It makes for a much more robust experience. So thank you, everybody. We'll be circulating follow-up information by email.